In the annals of scientific discovery, few achievements rival the magnitude of deciphering the human genome. This groundbreaking endeavor, akin to mapping the very essence of our being, has ushered in a new era of understanding human health, disease, and evolution. At the heart of this revolution lies a technique called Sanger sequencing, a method that transformed the esoteric world of DNA into a readable code. Developed by Frederick Sanger in the 1970s, this ingenious method provided scientists with the tools to decipher the precise order of nucleotides: adenine, A, thymine, T, guanine, G, and cytosine, C, that constitute our genetic blueprint. This knowledge, once hidden within the intricate double helix of DNA, became accessible, opening up unprecedented avenues for scientific exploration. Imagine a vast library filled with volumes upon volumes of information, each page inscribed with an enigmatic language. Sanger sequencing provided the key to unlocking this library, allowing us to read, interpret, and understand the language of life itself. This breakthrough paved the way for identifying genes associated with diseases, developing targeted therapies, and tracing the intricate tapestry of human ancestry and evolution. The ability to read our genetic code has not only revolutionized medicine, but has also profoundly impacted fields as diverse as forensics, agriculture, and conservation. The journey of Sanger sequencing begins with a seemingly simple yet crucial step: separating the two strands of the DNA double helix. This process, known as denaturation, disrupts the hydrogen bonds that hold the complementary base pairs A with T and G with C together. Imagine a zipper being unzipped, separating the two interlocking rows of teeth. To achieve this separation, scientists raise the temperature of the DNA solution. Heat provides the energy needed to overcome the attractive forces between the base pairs, causing the double helix to unwind and separate into two single strands. This process is akin to melting, where heat disrupts the ordered structure of a solid, transforming it into a more fluid state. Once the DNA strands are separated, they become accessible for the next crucial step: primer attachment. These short, single-stranded DNA segments serve as starting points for DNA synthesis, guiding the enzyme responsible for building new DNA strands. The choice of primers is crucial, as they determine the specific region of DNA that will be sequenced. By selecting primers that bind to flanking regions of the target sequence, scientists can ensure that only the desired portion of the DNA is amplified and analyzed. With the DNA strands separated, the stage is set for the next critical step: primer attachment. These short, single-stranded DNA segments act as starting points for DNA synthesis, guiding the enzyme responsible for building new DNA strands to the specific region of interest. Imagine them as bookmarks, flagging the precise section of a vast library shelf that needs to be read. The primers are designed to be complementary to the flanking regions of the target DNA sequence. This complementarity ensures that they bind specifically to the desired location, flanking the segment of DNA that scientists want to sequence. Once attached, they serve as initiation points for the enzyme DNA polymerase, which plays a central role in replicating DNA. DNA polymerase is the molecular workhorse responsible for building new DNA strands. It reads the sequence of the template strand and adds complementary nucleotides one by one, following the base pairing rules, A with T and G with C. The primer provides a crucial starting point for DNA polymerase, allowing it to latch onto the template strand and begin the process of DNA synthesis. Without primers, DNA polymerase would be like a painter without a canvas, unable to initiate the process of replication. With primers in place, the stage is set for the star of the Sanger sequencing show, DNA polymerase. This remarkable enzyme orchestrates the synthesis of new DNA strands, meticulously adding complementary nucleotides one by one, guided by the template strand and the faithful base pairing rules. Imagine a microscopic construction worker diligently assembling a new wall brick by brick, following a blueprint. The process begins as DNA polymerase encounters the primer attached to the template strand. Recognizing this as the starting point, The enzyme latches onto the primer and begins reading the sequence of the template strand. As it moves along the template, it selects free-floating nucleotides from the surrounding solution, matching them to their complementary bases on the template strand. For every adenine, A, on the template, DNA polymerase adds a thymine, T, to the newly synthesized strand. For every guanine, G, it adds a cytosine, C, and so on. This precise matching ensures that the newly synthesized strand is an exact complementary copy of the template strand. As DNA polymerase continues its journey along the template strand, it adds nucleotides one by one, extending the new DNA strand. This process, known as elongation, continues until a special type of nucleotide, called a terminator, is incorporated, signaling the end of DNA synthesis for that particular strand.